DNA replication and protein synthesis.
Helicase comes in first during DNA replication. Helicase is responsible for separating the gene in two. Then the primase will begin priming the worksite, making it easy for the polymerase to do their job.
Primase is a kind of primer. It primes the worksite in order for the polymerase to be able to do their job properly and efficiently.
But wait, one of the phosphate backbones is from 3' to 5', which means it's the lagging strand, instead of the correct 5' to 3', which we call the leading strand. This makes things harder for DNA polymerase to build the correct sequence of amino acids. Oh no, looks like we need some more primer. We need to make Okazaki fragments so everything can work.
Now comes the ligase to glue all the replicated DNA together.
Now we have two identical replications of DNA.
Now we're two!
We are the same! DNA transcription.
Transcription begins with initiation, where DNA is made into mRNA.
Now we can start transcription, which is making DNA into mRNA. First step is initiation. RNA polymerase comes along to separate the DNA strands at a special starting point called the promoter. Now we can bond the RNA nucleotides with their base pair in the DNA template. DNA polymerase and RNA polymerase are similar because they can only assemble the polynucleotide in the 5' to 3' direction. Fun fact! RNA does not need to have a primer to start their building process.
Now we can begin elongation.
As RNA polymerase moves down the DNA, it unwinds 10 to 20 bases at a time. That's so many!
There are 3 rules to elongation. Rule number 1, add complementary bases. Rule number 2, uracil replaces thymine.
Rule number 3, the double helix reforms and mRNA says bye-bye.
When a protein is in high demand, RNA polymers can make several of the same gene at the same time, producing several mRNAs. The RNA polymerase ability to do this can stop this from happening.
Fun fact! There are 3 types of RNA transcribed in the nucleus. There is messenger RNA, transfer RNA, and ribosomal RNA.
Now that we've finished elongation, we can begin the final step in transcription, which is termination.
Transcription stops when RNA polymerase comes to a terminating sequence.
mRNA is kicked out by the RNA polymers through a nuclear pore and then is sent to the cytoplasm, which is then translated into an amino acid sequence.
The whole transcription process makes one gene.
Yay, transcription is done. Now we can move on to translation. Translation is when mRNA gets turned into protein.
Translation. This step converts nucleic acid to polypeptides.
This occurs with ribosomes. For those who don't know, ribosomes are organelles that don't have a membrane. They are composed of protein and RNA. Ribosomes are found in the ER or hanging out together in the cytoplasm.
Ribosomes read from a triplet code called codons. This is a universal language for nucleic acid. This language is not able to be read by amino acids, so a cell must translate this language and build polypeptides.
Boo boo beep beep do 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 do. The dog came home last night.
I am the translator. I am tRNA.
The ribosome adds the amino acid, which is brought by the tRNA, to one end of the polypeptide chain. This means the polypeptide chain will grow.
At the end of the tRNA, there is a group of 3 nucleotides called the anticodon. The anticodon has the job of binding the nucleotides of a codon together by complementary base pairing.
Now that we've talked about the basics of translation, let's get into more detail.
First stop is translation initiation.
Initiation starts when ribosomes line up with mRNA at the AUG which is known as the start codon.
Now the tRNA lines up with the right anticodon and the right amino acid on the codon of mRNA. The ribosome moves along the mRNA one codon at a time. The tRNA lines up with the other codon. The ribosome must check that the anticodon/codon match up before it can catalyze a peptide bond between the amino acids.
Now we begin translation elongation.
The ribosomes continue reading the codons and matching them to the correct amino acids.
The enzyme on the ribosome will begin catalyzing peptide bonds.
The chain grows and grows one amino acid at a time.
Yay, we're on the last step of translation. This step is called termination.
Termination can begin once the ribosome has read the terminate codon, UAG, then it will stop making amino acids. It will then release the protein, and the protein will fold into its designated shape.
After the translation has been completed, mRNA will disintegrate. Now we will show you how to use the codon chart and what that does.
First, we start with DNA.
Now we have mRNA, where we have complementary base pairing and the T is replaced with a U.
Follow the codon chart to get the same amino acids that we have.
Use this codon chart starting at the middle and working outwards to get the same amino acids we did.